Okay, so let's get started. So first, for a couple of minutes, if you have any question, let's take those.
So could you discuss second type of epitopes, that is linear epitope again? Like yesterday, it was not clear. So
Okay. So basically, linear epitopes, or the other name of linear epitope is sequential epitope. Okay, so you have basically a sequence of amino acid in a protein, right? Amino acid at position 1, 2, 3, 4, 5, 6, 7, and so on and so forth, right? Now, so if you have a contiguous sequence which gives rise to an epitope which can stimulate B cells, these are called linear epitopes or sequential epitope. Okay, and these were some of the examples. Okay, if you look at the N terminus and C terminus of this globulin of whale sperm, right? So you have, for example, one epitope which is concentrated here, 15 to 21. Another epitope actually is concentrated here, 56 to 62. So this is a sequential, this is actually a sequence starting from 56, then 57, 58, 59, right? 60, 61, 62. So this region actually constitute one epitope. So these are called sequential or linear epitopes. In other slide, so here is the example of an epitope which is generated when a protein has acquired certain structure. So it's not a linear sequence anymore. Okay. For example, if it has acquired something like as this, then you create a shape which can be bound by the paratope. Right. Now, if you open up this sequence and you make it linearized, then this antibody does not bind. Okay, so it's not like what is shown here with blue dots is all the amino acids, and starting from 64 to 80 amino acid, you have one epitope, but that does not say that this epitope actually is critically involving every single amino acid that you have. Maybe it's present in this region, and this region will only form this epitope once this has formed the disulfide bond, right? If you, for example, treat it with DTT, diethioethritol, or 2-mercaptoethanol, 2-me, right? These are reducing agents. So they will break these disulfide bond, and once those disulfide bonds are broken, so then the antibody does not actually bind to this protein. So that means these epitopes are not in a linear sequence, because with disulfide bond breakage, so you actually create a linear structure. And on that linear structure, so you don't bind the antibodies that were specific to the folded protein. Okay, so this epitope would then be considered as non-sequential or conformational epitope. Yes?
got it, sir. Thank you.
If no more questions, so let's keep going. Okay, so we also talked about the other properties. One was basically these are hydrophilic, accessible, they can exist in sequential and non-sequential form, and these are two of the examples I showed you, right? And then third property is these B cell epitopes actually are present in the flexible region of a protein. And the region is because of the inherent flexibility in that region, they can actually snugly fit into the paratope region of the antibody. Okay. Fourth one was that these can be overlapping, that it's not like 1 to 10 amino acid constitute 1 and the next epitope has to start from 11. It can actually start from 7 and then it can go up to 10 or 12 or 14, whatever. So there is an overlapping. Basically, these epitopes can actually come from within the overlapping region of the amino acid sequence that you have. The other thing that we talked about was that on a given protein, there are only certain sequences or certain regions which are recognized more and others are not recognized that well. The recognized means recognized by B cell receptor, right? Or and eventually they are bound with the antibody, which is actually formed by activation of these B cells. And the preceding step in activation of B cell is binding of the BCR with these epitopes. Okay. Now, those epitopes which bind or which induce a good antibody response or a T cell response, because we are talking about B cell here, so let's only talk about B cells. Okay. Those epitopes or those regions on a protein molecule which interact well and induce a good magnitude of response against that region in terms of the antibody production. So they are called immunodominant, and those epitopes which are not so well recognized, they are called either subdominant or non-immunodominant. Okay. And then we end up saying that aim of any vaccination strategy is to identify immunodominant epitopes and include those epitopes in the vaccine formulation. Now, how do you identify that these are immunodominant epitopes, these are not? There are different approaches. You can go through a forward approach, or you can take a reverse vaccinology approach. And a little bit of reverse vaccinology we talked about when we had this history of different vaccines, right? So essentially what it means is forward approach is, so you are sampling, your first step actually is sampling of tissues or serum, right? Or a lymph node from infected or immunized individuals. And then you go back and test that in this sample, so what epitope specific response have you accumulated? This is forward. Okay, so you basically go from one step. If someone is infected, so you actually collect serum or tissue samples from lymph nodes, and then you find out which cells are reactive to what epitope. This is forward approach or forward vaccinology. Reverse vaccinology is you start with the sequence of the microbe or that antigen. And these days, finding a sequence of any antigen or any microbe is maximally a 1-week job, right? So you know that this virus, coronavirus, it came in 2019, and within rather a month's time, so this whole sequence was published in and was available in public database within months time, right? Now, starting from there, if you actually can, this is actually a nucleotide sequence. You can predict amino acid sequence from that, and once you have the amino acid sequence, then you can identify epitope. Yeah, eventually you have to establish the immunogenicity using experimental approach. But this approach where you start with the nucleic acid sequence is called reverse vaccinology to identify immunogenic epitopes. All right. So is that clear? What is a forward approach and what is a reverse approach for identifying immunodominant or subdominant or non-immunodominant epitopes? In one case, you start with the sample first, and sample is collected from infected individuals. In another approach, you start with the nucleic acid sequence, and then you go back and find out. First, you perform a prediction, and then based on prediction, then whatever you have selected, you establish the immunogenicity. All right. So after that, so we are going to talk about actually. So now you already know that these are the properties of your B cells epitope, right? In essence, these are actually four. They are hydrophilic. They are predominantly present in flexible region, exposed onto the protein. They can be immunodominant, right? Can be linear, conformational. The other name is basically sequential or non-sequential, right? So after that, basically, if you are talking about the phases of immune response, okay, so essentially it involves starting from naive T cells or naive B cells. These naive B cells are formed in bone marrow, and once they have gone through these processes, which make sure that you don't have autoreactive cells coming out of bone marrow. So these cells then populate periphery. In periphery, they recognize these antigens. They become activated, right? The activated B cells are also called as plasma cells. These plasma cells make antibodies. Antibodies help neutralize this microbe or these antigen that was actually injected, right? And a pool of these effector cells or plasma cells that you generated from naive cells then become long-term memory cells and stay in your system almost for the rest of your lives, right? And same is the case with T cells. You start with naive T cells. They also start their journey from bone marrow. From bone marrow, they get to thymus. In thymus, they undergo this process of negative-positive selection. Once it is made sure that you have not selected any autoreactive T cells, these cells then come to secondary lymphoid organs. In secondary lymphoid organ, they sense the presence of these antigenic epitopes loaded onto MSC class 1 or class 2, depending on whether these are CD4 T cells or cytotoxic T cells, right? And then by receiving two additional signals, co-stimulatory and the cytokines in the milieu, so these cells become activated. We call these cells as effector cells. There is actually a burst in the number, increase in the number of these effector T cells. They try to control infection by killing the target cells. Target cell means the infected cells or transformed cells. And a pool of those cells, which rarely account for 5 to 10%, then become memory T cells. And these memory T cells also come in different flavors. They can be found in circulation. They can actually be also present in your tissue that we call as tissue-resident memory cells or circulatory memory T cells, right? Central or peripheral. So there are different nomenclatures, but essential property is they remain in the system and they can be recalled following exposure to the same antigen very quickly and very rapidly, and within a span of couple of days, their number become too much that they can control this infection very, very efficiently. You don't even know that you got the second infection, and by the time, so your memory cells have already been reactivated to become effector cells, and the effector cells have controlled this infection. And same is the case with antibody forming cells. Memory B cells form these antibodies much more efficiently than naive cells. And the only difference is because these naive cells have not really seen antigen in the right context, but memory B cells were generated after seeing this antigen. So that is, by definition, we call them as memory cells. Okay, they can remember the presence of that antigen. So is that clear? That this is how this sequence of event take place. All right, so more or less this is what we are going to talk about, but this is actually one of the theory which is accepted, which is actually highly accepted in the field of immunology, and it was given or it was actually proposed by F.M. Burnett way back in 1951, rather in 50s or 40s and 50s. And it was not one of the greatest paper that was written. So you would like to say that if somebody, someone has proposed a theory which nobody is able to refute until today, so that has to have some value, right? And if there is some value associated with this theory, obviously this should find its place in one of those ZWIS journals like Nature Cell Science, right? Apparently that was not the case with this clonal selection theory or clonal selection hypothesis given by F.M. Burnett, and it was published in Australian Journal of Science. It's like Current Science that we have here in India, which has actually an impact factor of less than 1, and at that time it didn't have any, right? But once this theory was proposed and evidence came that this actually is true and nobody was able to refute this theory, F.M. Burnett was recognized for his contribution to propose this theory, and he was given Nobel in 1950s, 51, I guess, right? There were many more theories. Briefly, we'll talk about those maybe next class or in one of those classes, right? But all those theories did not hold true. So they somehow were not able to explain the principle of adaptive immunity. But this theory is still considered as the gold standard. And this is how actually immune response work, right? So you actually see these different postulates of the theory. Okay, the first postulate is each lymphocyte bears a single type of receptor, right? And receptor is basically on the surface. And these receptors, whatever the number of receptors you have, the specificity of each of those receptors is 1. It has a unique specificity. So it's not like, so, and when we talk about the surface receptor on lymphocyte, rather antigen receptor, so we are talking actually about the membrane-bound antibody or membrane-associated T-cell receptor. Okay, so the first postulate was that each lymphocyte bears a single type of receptor with a unique antigen specificity. Even if you have 5,000 or 10,000 or 1 million of those receptors on the surface, each one of them will have one particular specificity, the same specificity. Okay, the second postulate was that interaction between a foreign molecule, foreign organic molecule, and a lymphocyte receptor. Again, receptor we are talking about is the antigen receptor, the BCR in this case, or TCR on T cells, right? Interaction between this foreign molecule and the lymphocyte receptor capable of binding to it with high affinity leads to lymphocyte activation. See, when it was basically when these See, the aim was to explain how actually you can generate so many different types of specificities that you can recognize each and every antigen. Is it that once you are immunized, you start developing the specificities, or you already have those preformed specificities on the surface of these cells, and then you choose one of those, or maybe all these specificities are present on one lymphocyte, right? And then you have multiple of those lymphocytes, and whenever you actually encounter an antigen, that antigen bind to one of those specificities out of many present on one cell, and then activate these cells to produce only that kind of antibody. So there were so many different theories that were floating around at that time, but nothing was, or none of those were able to conclusively suggest that this is a logical way how immune response might be induced. And this clonal selection theory was able to provide with authority that this is how T cells or B cell responses are induced. Okay, and we are talking about the postulates of this theory. The first one was that each lymphocyte bear unique antigen specificity on their surface via their antigen receptors. Now, if you have the right interaction between a foreign molecule and the lymphocyte receptor to which this foreign molecule can bind, thus this is going to activate this particular lymphocyte. This activated lymphocyte will be differentiated into effector cells. Okay, now all these effector cells will come from an activated lymphocyte and will bear identical receptor specificity, okay, which actually were present on the parent population or parent cell. Okay, now lymphocyte bearing receptor specific for ubiquitous self molecules are deleted, so they are not actually present in the periphery. Okay, they are deleted at an early stage in lymphoid organs during their development process, and we did say that these developmental processes take place at different sites for T cells and B cells. For T cells, these are occurring in thymus, and for B cells, they are occurring in bone marrow. Okay. Now, essentially, what these processes will make sure is that you do not have any autoreactive cells, or basically a pool of these autoreactive cells in your peripheral circulation or peripheral lymphoid organs. So this is the fourth hypothesis or fourth postulate of this theory. Although we know that this is not absolute. Okay, the fourth point is not absolute, but these 3 points are absolute. Okay, nobody is able to actually contest that this actually is not true, right? This will be more clear in this next slide. So lymphocytes activated by antigen give rise to clones of antigen-specific effector cells that mediate adaptive immune responses. This actually is the essence of the theory. Okay. The name of the theory is clonal selection theory, right? So let's start from 1. Okay. Now, what is shown here in this panel is a single progenitor cell give rise to a large number of lymphocytes, each with a different specificity. Okay, you have the progenitor cell. Now, this progenitor cell will differentiate into these different cells, and at this time point, the specificity of the receptor is decided. This is actually the developmental stage. This is not during the activation stage. This is when these cells actually develop in primary lymphoid organs. You start from progenitor lymphoid cells, they give rise to the cells of different antigen specificity. And this ensures, okay, all these specificities are there. So they have already been generated, and they are generated in a very random fashion. Now, if these are randomly selected, so it is quite possible that some of these clones might react to your own antigen, right? And that is taken care of during this process. Okay, so the next step is removal of potentially self-reactive immature lymphocytes by clonal deletion. There are two processes here. One is positive selection, one is negative selection. But eventually, so we call actually it as the clonal deletion. Those clones which are reactive to your own antigens. For example, this one was reactive to pancreatic antigens. This one was reactive to your skin antigens. This one was reactive to, I don't know, maybe your kidney antigens. This one was reactive to your liver antigens. All those clones of lymphocytes are deleted by the process of clonal deletion. And then you are left with only those that do not react with your own antigens. Is that clear? That autoreactive cells are deleted. The process is called clonal deletion. Okay, you are left with a pool of non-autoreactive cells, okay, which become then mature and they populate peripheral lymphoid organs. In other words, secondary lymphoid organs, and they are also present in circulation. You are left with these three clones. It's not three, it's just a cartoon to show you still have billions and billions of specificities remaining. But those clones which were potentially autoreactive are deleted. Right? Once these are present in periphery and they are waiting for this antigen to come and activate them, and once this antigen come and basically you have the right signal given to your T cells or B cells, so there is a proliferation, there is a burst of those lymphocytes of only this kind of specificity, because this is decided by the foreign antigen interacting with this specific receptor. This is not going to basically stimulate the other cells, right? This is going to stimulate only this particular cell, and then you are left with so many different types of so many different numbers, okay, of this particular specificity. You are actually expanding the number of those clones, and what the purpose of expanding those number of clones is because you are encountered with this particular antigen, and when this antigen somehow is present, so this needs to be removed from this system, and this is basically how it carries out, either by the surface recognition of this antigen by your T cells, or by the secreted product. In other words, basically these are the secreted receptors, and now the receptor which is secreted out and which is tethered to the membrane are different, which is actually true for B cells, not for T cells. In T cells, TCR is never secreted. Okay, by recognition through TCR, so these cells make something else which is responsible for eliminating this antigens or microbes or cancer cells, right? But for B cells, the same specificity of the B cell receptor is retained. There are subtle changes in the transmembrane and the cytosolic portion of this receptor, so those are removed, and only the secreted antibody molecule will then bind to these antigens and they try to neutralize it. So these are basically the steps of clonal selection theory. If something is not clear, ask it before we go to the architecture of the receptors. It's clear? Nikita,
Yes, sir.
is it clear?
Yes, sir.
So any
But
point that needs to be
I was thinking that
I'm not able to hear you. Looks like there is some problem with the connection. So it's okay. So anybody else? Pranav? Pranav,
Yes, sir. It's clear.
it's clear. Okay. All right. So go through this theory again. So and if something is not clear, so we'll discuss maybe in next week's class if we have one. All right. So this is the composition of basically the antibody molecule or the T cell receptor, and you could see essentially we are talking about the antigen receptor. Okay. And you would recognize that antigen receptor is nothing but this antibody molecule, right? So it consists of this variable region, right? And you have these different regions, different domains, okay? And then you have the constant region. Constant region actually is marked with this blue, okay? This blue, this blue, this blue. Variable region is with what color, whatever it's, okay? So in this portion, you have the variable region, and in this portion, you have the constant region. And you actually can see 4 chains. 2 are identical, and these 2 are identical. Okay. So these are called light chains. These are called heavy chains. Heavy chains have this disulfide bond. Heavy and light chain also have this disulfide bond, but you don't find light chain and light chain combined, right? So this is a Y-shaped structure. At the tip of this Y-shaped structure, you have the antigen binding portion that we call as paratope. The constant region actually is having some extension, which is not shown in here, which actually traverses through the membrane of B cell, and then it has a short cytoplasmic tail, right? There's something in the chat box. So what was it? Can somebody read and
So Nikita is asking, how do B cells neutralize the antigen after binding?
well, okay, so B cells once get activated, so they are going to release a lot of these antibodies. They are going to secrete out a lot of these antibodies, and these antibodies will bind to the antigen, and then they will neutralize it. The portion which actually is tethered to the membrane and the cytosol, they are cleaved off by a process that we'll talk about during the differentiation of B cells. Okay. Now this is BCR, which is having the identical structure to your antibody molecule, but in BCR, the additional thing that you also have is a domain which actually traverses through the membrane, and then you have a short cytoplasmic tail. Okay. On the other side, so the structure of TCR is shown. You have two chains, alpha and beta. You have the variable region, and then you have the constant region. And then alpha and beta chain actually are hooked together with this disulfide bond. They have this transmembrane portion and short cytoplasmic tail. And there is a significance of having this short cytoplasmic tail, because this alone cannot signal. So you have to have some accessory molecules, some adapter molecules, so which actually also are part of this TCR or BCR complex. We'll talk about those things at some time point. because eventually, if it is a receptor, it has to transduce signaling in the cells. And for signaling events to be transduced, so you require many more components here. But the recognition is brought about by only this portion, alpha and beta for TCR, and heavy and light chains for the alpha and beta for the TCR, and heavy and light chains for the TCR, BCR. Don't get confused. So this is BCR, this is TCR. Okay, so let's now talk about the basic structure of antibodies. So now we are actually starting with the antibodies. Okay, and these are different things that we are going to discuss in antibodies. The basic structure of antibody, antigen binding sites, antibody mediated effector functions, classes of different types of antibodies, and what are the biological activities associated with them. Then we are also going to talk about the structure of the BCR. Okay, and we know that BCR actually is part of this immunoglobulin superfamily member. So then we are very briefly going to discuss what are the other molecules which are part of this immunoglobulin superfamily, and eventually we'll end up this session. This session means the section on antibodies with monoclonal antibody. How you can actually generate these monoclonal antibodies, and what is the process, and what does it require. Okay, the steps remain same. Okay, so and this actually you have to understand very well before actually you can start understanding about the antibodies, right? You start with stem cells. In stem cells, there is this process of gene rearrangement. We'll discuss that. So what are different segments and so on and so forth, and how they are combined, right? So gene rearrangement will give rise to this specificities of different types, as are shown here by different colors. Yellow is having one antigen specificity, which is actually brought about by the exposed receptor. This pinkish cell will have a different second antigen specificity. Then you have blue, third, and fourth, and fifth, and 1 millionth, and 1 billionth. All those kind of specificities are generated by this process that we call as gene rearrangement. Okay. All these processes actually are taking place starting from stem cell to the formation of these different antigen specificities in bone marrow, right? Once mature B cells are formed in bone marrow, so they will then come to peripheral lymphoid tissues. Now, in peripheral lymphoid tissues, they keep on homing from one side to the other, one place to the other, and they actually are ready to recognize any antigen you are exposed to. For example, out of those many specificities, so you encounter this antigen number 2, which can stimulate this clone 2 of your mature B cell. Now, this cell will start getting activated, the activation process, undergoing the activation process. It will basically form many similar clones which have the same antigen specificity. Now, basically, from one cell you are going to give, from this one cell you are going to give rise 2 cells, from 2 4, from 4 8, and you keep on going for next 1 week or 2 weeks time. And starting from this one cell, you can actually make billions of these different antigen specific cells, or maybe millions. Okay, millions to billions, so in that range, following this antigen exposure. Once these cells are activated, so they're going to produce antibodies. Some will, as I said, become memory cells. They are going to re-respond to the same antigen, because it's not like that once you have gotten one infection, you will never get it again. Because if infection is present, so you'll keep on encountering it, right? So immune system is so smart that it generates this immunological memory for that antigen, so that when you actually come up with this second time infection with the same pathogen, so these cells are recruited in the response very quickly. They make these antibodies and you try to neutralize whatever microbe you are encountered with, right? So this is basically how the process of B cell activation take place. I've not really talked about the events as such, but this is a schematic. Start from stem cells undergo gene rearrangement, give rise to different antigen specificities. Different antigen specificities, if are selected for appropriate specificity, so that you don't have the autoreactive cells, you have only the foreign antigen reactive cells. They basically then come to periphery in your secondary lymphoid organs and circulation, and they sense the presence of this antigen, they expand in number, and they produce these antibodies. Those antibodies are going to neutralize the antigen, right? All right, so this is one process where basically the interaction of different cells is shown, just because B cells do not work in isolation as such, okay? Initially, they can get activated only by seeing the antigen, but for their downstream processes, so that you can start making high affinity antibodies, you can generate plasma cells which produce better quality antibodies, and then formation of memory cells. All these processes require cell-to-cell interaction. Particularly, the help is provided by T cells of CD4 lineage. Okay, so broadly, so basically you are looking at this cell, B cell, right? B cells have on surface of on their surface these B cell receptors. These B cell receptors see or sense the presence of this antigen in unprocessed form. Once you generate these complexes on surface, so this antibody molecule along with the antigen is internalized, and then these B cells can actually also cut down this antigen into small pieces to generate those peptides. Those peptides can be loaded onto MSC class 2. You know, B cells are also actually can serve as antigen presenting cells, right? B cells, macrophages, dendritic cells. These are three professional antigen presenting cells. And basically this process is telling you how B cells can serve as antigen presenting cells. Okay. They express a lot of MSC class 2, right? Internalization of antigen is effected through B cell receptor. B cell receptor bind to antigen, internalized, cut it into pieces. Those pieces actually are the class 2 derived epitopes or class 1 derived epitopes. But in order for B cells to get activated, so this peptide is loaded onto MSC class 2. This is going to activate your CD4 cells. CD4 cells, once activated, will produce the cytokines. The cytokine receptors are present on your B cells, and these cytokines signaling along with this antigen receptor signaling is going to make these cells into plasma cells, which can make good quality of antibody response, right? And once activated, some of these cells also become memory cells. Now, memory cells, by sensing the presence of the same antigen, can also become plasma cells. Now, there is a question mark here because many of the events are not known. Okay, some people say that plasma cells can also stay up for a long time, but normally in the thinking in the field actually is that plasma cells are short-lived cells. Their purpose is to spit out these antibodies and they die out. But theories are being given and revised and re-revised, and so all those things are happening. So that is why this question mark. But is that clear? Where basically T cells come into picture for activation of B cells? And this process actually is taking place at a particular site in lymph node, in secondary lymph node, that we call as germinal center. And the process is called germinal center reaction, GCR. Okay, this has nothing to do with the germinal cells, basically in the reproductive tract. So this is for the activation of your B cells. This is a site, this is a location in secondary lymphoid organs, which is responsible for making these antibody producing cells or activating these antibody producing cells. All right. Yeah, so actually, now we are going to actually talk about some historical data, basically where for the first time when antibodies were described, and basically how did they identify that these molecules, these proteinaceous molecules that you have in serum, and they can be actually induced following immunization or vaccination. And then following vaccination, so you actually generate these protein molecules, and these are the properties of those protein molecules, biochemical properties. And they are responsible for binding to the antigen that was injected in animals for inducing their response, and then they can actually carry out their effector function. Okay, so essentially, the initial studies were done by these two scientists, Tiselius and Cabot, Alvin Cabot and Tiselius. Okay. And even today, the sequence of amino acid that you have in both light chain and heavy chain of your antibody molecule is called Cabot numbering. Okay, because they somehow found some similarity that at a particular position, if you have only this particular amino acid, so this should be numbered as whatever. Okay, so there is a way of defining the composition of antibody molecules by Cabot numbering, and this Cabot actually is coming from the name of the scientist. Okay, these experiments were done way back in 1930s. Now think about it. So what kind of facilities were available around that time, 1930s? Okay, so people actually used to work on these slides, and they somehow put this electricity. Electricity was also actually newly a new thing around that time. Okay, you connect these two electrodes and then try to basically see what kind of characteristics, whether it's a positively charged molecule, negatively charged molecule. So all those things actually were defined by these scientists. Okay, the procedure. So what was the procedure? Before that, actually, they discovered that the gamma globulin fraction of serum. Do you know what serum is? We did talk about quite elaborately in one of these classes, serum and plasma. This is the soluble fraction of your blood. If it is removed of all the cells, all the blood cells, so then whatever you are left with is plasma. And if you remove from plasma these proteins which are responsible for blood clotting, so then you generate serum. Okay, in serum, so you have a lot of these antibodies. Serum actually is highly enriched in the antibodies. What they showed was that in serum, so you have a fraction which they defined as being gamma globulin, and that gamma globulin region actually is rich in antibodies. Okay, gamma globulin. So they were actually different types of globulins, alpha, beta, and gamma. So they were actually defined based on where they would migrate when you apply electric current. Right? So now you also, if you are collecting serum have different kinds of antigen specificities, but you have to actually enrich that serum sample with a lot of antibodies specific to a particular antigen. only then you can define, you can actually show the binding and so on and so forth, right? So what they did was, so they immunized rabbits with this model antigen, chicken of albumin. Okay, so this is egg of albumin. So the white fraction of the egg that you have. Okay, so basically that fraction is called, that has this protein that we call as ovalbumin, right? This actually is serving as a model antigen, and almost it will not be a basically hyperbolic statement if I say that almost every single phenomena, maybe almost 70 to 80% phenomena that you know of today in immunology actually have come from this protein. Okay, chicken of albumin. This actually serves as the model antigen. So why actually this serves as the model antigen? So maybe we can talk about it at some time point. So it has T cell epitope, it has B cell epitope, it is available in large abundance, and you can inject it. It's innocuous, it's very cheap. When you generate antibodies, you can actually measure the specificity of those antibodies. There are many features which are actually good for this antigen, right? So they immunized animals, the rabbits, with this chicken of albumin. Okay, so this was actually serving as the model antigen. Collected serum from these immunized rabbits. Okay, and they defined, they actually, when they were doing these kind of binding assays, what those assays were, they were like immunoprecipitation kind of assay. So you have your antigen, you basically add it into this immune serum that you collected from rabbits, which are initially injected with this proteinaceous antigen. Okay, and then whatever this stuff will bind to the antigen, so that will become heavier. And once it becomes heavier, it will tend to settle in the bottom. You can centrifuge, and all the precipitates, the bound form, will be settled in the bottom of this tube. And whatever the fraction which is unbound, so you collect it, and then you find out what has changed in the unbound fraction versus the whole serum. Okay, and this is the kind of experiment they did. And the experiment actually is called electrophoresis. Rather immunoelectrophoresis. That will come in the next slide, but for the time being, just look at this thing here. You have two lines here. One is this bluish line, and another is black. Black line is where you had this serum. Serum was collected from animals which are immunized with chicken albumin, right? And in this serum, so they added this antigen, waited for some time, centrifuged it, and collected only the supernatant. And they basically ran it onto the, basically they resolved it under the influence of this electric field. How you can generate this process? So this actually is called, so you have this slide, for example. On this slide, so you make a thin layer of this agarose gel, right? Few mm, and then you cut basically a punch in the gel. You can actually make an area where you can actually load some sample, right? So all of you know about SDS page and electrophoresis and agarose gel electrophoresis, right? So if you have some this agarose gel solidified onto the surface of this slide, you make a punch. So you make a basically area where you can load sample and apply electric current, and then let all the components resolve depending on their charge. If it is negatively charged, it will move towards positive electrode, and if it is positively charged, it will move towards negative electric electrode, right? Depending on that, you can actually separate different fractions. Once those are separated, you can actually cut the piece of gel and then quantify how much protein is present. Okay, by taking absorbance at 280 nanometers. This actually is shown on this y-axis here. And on x-axis, you are showing that, for example, let's say if you have if you have loaded your sample here, from here to here, so what is the distance in centimeters or millimeters? What is this distance up to this point, up to this point? Okay. Now it was defined that the farther from this loading sample towards the negative electrode, so you have the migration of this gamma globulin fraction. Then you have beta, then you have alpha, and albumin actually is coming in this region. And that is because of the charge present on albumin, alpha globulin, gamma globulin, and beta globulins, right? Now, what you see is that this serum from rabbit, which was not adsorbed, so it shows all other peaks. Every peak actually is present here. But the sample that was actually added with this chicle of albumin and precipitated, so it did not have a lot of protein in this fraction. So there is quite a huge difference in this region. So that means in serum, whatever component bound to the antigen, so that actually has the ability to interact with the antigen, right? And they then defined this as because this was actually running in the globular region, right? So they called it as gamma globulin fraction. And because it has immune reactivity against the antigen, they call it as immunoglobulin. Immunoglobulin actually migrate in this region. All other was not changed after adsorption. Albumin is not changing, alpha globulin is not changing, beta is not changing, but gamma globulin fraction is removed from the serum when it was added with the antigen. Is that clear? So you know that the nature of the antibody molecule actually is gamma, because this was migrated in gamma region. It was a globular protein and it had immune reactivity against the antigen. So immunoglobulin. Okay, I think we have to actually stop it now. We'll continue tomorrow.